You've probably all seen the pregnancy test commercial that says, first response tells you 5 days sooner. Or maybe you've seen their more recent ad, first response tells you 6 days sooner. Well, clearly pregnancy isn't the only example of the importance of early detection. What about diseases like Ebola, where the time between showing symptoms and death is only 14 days? Or prostate cancer, where patients typically present symptoms in such late stages that chemotherapy is no longer an option. See, the cancer starts as a single cell, but its biomarkers, or the proteins associated with that cancer, are undetectable until enough cancer cells are present to form a tumor. The goal of my research is to design biomarker sensors that are capable of detecting such low levels of biomarkers they could detect a single abnormal cell. But I'll get to my research in a second. Let's first talk about basic sensor design. How does a small molecule like a biomarker go about triggering a detectable event? Well, typically the biomarker binds to another molecule at the sensor surface, and this induces a measurable change, like a change in voltage or a change in color. The molecule that I use at the sensor surface is called an aptamer. Aptamers are short DNA sequences, just like the DNA in our own cells, that bind to biomarkers. And they're great for sensing applications for two reasons. First, aptamers only bind to certain types of biomarkers. And this eliminates a familiar problem in sensing, the false positive. And second, aptamers undergo a structural change whenever they bind to the biomarker. The aptamer starts in a folded state, and when it binds to the biomarker, it becomes an extended structure. We can monitor this change with a green dye attached to the aptamer that only turns on in the extended form. Now, the primary aim of my research is to lower the detection limit of these aptamer sensors by enhancing a single property, and that's the attraction between the aptamer and the biomarker. To do this, I tested a simple hypothesis that if the aptamer is easier for the biomarker to locate by being exposed, then the two will interact more frequently and more binding events will occur. This is exactly the trend that I observed in my data. When the aptamer was exposed by being positioned at the top of the folded structure, the biomarker was 10 times more likely to bind than when the aptamer was hidden at the base of the sensor. With this simple modification, I was able to detect biomarker concentrations that were 5,000 times lower than previous aptamer sensors. So, to sum up, are we at the single abnormal cell detection level? Not quite, but we're getting close. Lowering detection limits by 5,000 times means potentially detecting Ebola up to 2 days sooner, or diagnosing cancer months in advance, or maybe even knowing you're pregnant up to 7 days sooner. Thank you.